Okay, so welcome to this first video in the playlist on lipid modification of proteins. Okay, so in this video we're going to introduce the topic by looking at the main types of lipid modification of proteins. Okay, so basically the idea is that we are going to be sticking lipid molecules onto proteins, okay? So fatty molecules. Lipids, remember, are another name, they're the proper name for fatty molecules. Molecules which have very long chain, long aliphatic chains, basically. Okay, right. So, the structure for this video then, we're going to start off by looking at the three main types of lipid modification of proteins, and then what we'll do is go through each of them in turn. Okay, so let's start off by looking at the three main types of modification to proteins. Okay, so remember, a protein is a polypeptide, okay? We'll forget about the tertiary structure for now. Obviously, proteins can be made up of multiple polypeptides all stuck together, but for now we'll just consider a protein as a polypeptide. Okay, so it's a polymer of amino acids, fundamentally, so it's amino acid after amino acid after amino acid after amino acid after amino acid, all joined together by peptide links, okay, which are these secondary amide bonds. Okay, and then the polypeptide folds up into a tertiary structure, okay, so the amino acids interact with one another and it folds up, okay. Right, so what we are now going to do is stick on lipid molecules onto the sides of the amino acids, basically, and therefore have lipid molecules attached to our protein. Okay, right, so when you have, let's show this, a lipid molecule attached to a protein, so I'll colour this in green here. So this is a lipid molecule in green here that's dangling off the side of our protein. Then the protein is known as a lipid-linked protein. Okay, so basically there are three different categories that you can divide lipid-linked proteins into. Okay, so let's do these now then. So category 1 is the prenylated proteins. Okay, so these are proteins which have had isoprenoid or prenyl groups stuck onto them, and I'll explain exactly what that means in a moment. Okay, so one of the categories is prenylated proteins. Okay, the second category then is the category of fatty acylated proteins. Okay, so these are proteins that have had just fatty acids stuck onto them. Okay, so acylated proteins, as they're called. Fatty acylated proteins. So when you add fatty acids onto proteins, you don't add the entire carboxylic acid group. In fact, you take off the alcohol group. So if I show you the structure of a fatty acid. Okay, so a fatty acid consists of a carboxylic acid group, then with a very long chain tail, basically, so a very long aliphatic tail here. Okay, and basically, when you add this onto a protein, you do not actually add the full carboxylic acid group. Instead, you take that alcohol group off, basically, and you add this group that's left that I'm now circulating in green here. Okay, so let me just complete that circle. Right, and this group is what's known as an acyl group. So basically, a fatty acid with the alcohol group of the carboxylic acid group taken off is then called an acyl group, and it's this acyl group that's going to be added on to the proteins, which is why this is called a fatty acylated protein. And I'm sorry that that Y is a bit confusing, but that's meant to be a Y there. So fatty acylated proteins, proteins which have had acyl groups of fatty acids stuck onto them. Okay, and we'll see two main types of fatty acylated proteins. Okay, and then the third category is what's known as glycosyl, phosphatidyl, inositol linked proteins. Okay, so glycosyl, and then it's phosphatidyl inositol Okay, and this is all one word. So glycosyl, phosphatidyl, and it's not going to fit in, but this should all be one word. Phosphatidyl, inositol, and then it's linked proteins. Now, there is a shorthand for glycosyl, phosphatidyl, inositol, and the shorthand for glycosyl, phosphatidyl, inositol is to abbreviate it to GPI. So glycosyl is abbreviated to G, phosphatidyl is abbreviated to P, and inositol is abbreviated to I. So these proteins would also be called GPI-linked proteins for glycosyl, phosphatidyl, inositol-linked proteins. Right, okay, so let's now work through these 3 different categories of lipid modification in order, basically. So we'll start with prenylated proteins and see what we're going to do, basically. Okay, so basically, let's start off with the structure which the prenyl groups are based on. So basically, prenylated proteins have had what are known as isoprenoid groups stuck onto them. Okay, and isoprenoid groups are also called prenyl groups. Okay, so you can either call them isoprenoid groups or you can call them prenyl groups. Okay, and prenyl groups are all based on the molecule isoprene. So we need to start by discussing the structure of the molecule isoprene. Then we'll see the 2 main examples of prenyl groups, which are namely farnesyl groups and geranyl groups. Okay, so let's start with the structure of isoprene. Now, I'll start by drawing out the molecular structure, and then we'll draw the skeletal structure, because when we draw these bigger groups, these farnesyl and geranyl groups, it becomes quite messy if you still use the molecular formula. It takes a long time and it looks nasty on the eye, so it's better to use the skeletal formula there. But initially, so that we have concrete understanding, we'll draw the molecular formula of isoprene. So, isoprene is a 4-carbon molecule where we have 2 double bonds, basically. And then off this carbon here, you then have a methyl group, so I'll stick that there. And off this carbon here, you have 2 hydrogens coming off here. Off this carbon, you'll have a hydrogen coming off there. And then off this carbon, you'll have a hydrogen here and a hydrogen here. So, this is the structure, the molecular formula for isoprene. Okay, now let's draw its skeletal formula. So its skeletal formula is shown like so. So remember, in skeletal formulae we do not show carbon atoms, they are implicitly shown by corners. Okay, so this double bond here represents this double bond here. Then we'll have a single bond here. And also note that in skeletal formulae you don't show hydrogens coming off carbon atoms. So that means we're not going to show an awful lot in this formula. Okay, so we've done 3 carbons now. There's 1 here, which is this one here. There's 1 here, which is this one here. And there's 1 here, which is this one here. And then we should need a final carbon, which will have a double bond between it and this 3rd carbon here. Now, we don't need to show any of these hydrogens coming off here, but we do need to show the methyl group, so we'll put that there. So this is the skeletal formula for isoprene. Now, basically you can polymerize isoprene together, okay, to make isoprenoid groups, and that's what these 2 molecules that we're going to stick onto proteins, namely the farnesyl group and the geranyl group, they're going to be polymers of isoprene, although they're going to be very short polymers. Farnesyl is basically going to be 3 of these isoprene molecules polymerized together, and geranyl is going to be 4 of them polymerized together. Okay, so let me show you how you're going to polymerize them together. So basically what you do is you break the second of these 2 bonds between this carbon and this carbon, and you also break the second of these 2 bonds between this carbon and this carbon. Now, covalent bonds consist of 2 electrons, 1 electron from each of the 2 members of the bond. So this covalent bond here that we've just broken, it will have 2 electrons in, and you can imagine giving one of those electrons back to this carbon and the other back to this carbon. Okay, similarly, when we break the second of the two bonds here, one electron will go back to this carbon and one back to this carbon. Now, what you're going to do is forge another covalent bond between this carbon and this carbon, because now this carbon has a free electron and this carbon also has a free electron, so they're going to join they're going to join forces basically and create a double bond there. So you're now going to get a double bond in the middle there. So this is a new bond, so I'll highlight it in pink here, so that's a new bond. Okay, the hydrogen remains off here, the methyl group remains off here. Okay, you now have single bonds here and here. These hydrogens I'll move out to the side, okay, like so. And now both of these carbons have free electrons, so they both have an electron that's unpaired, and they need a bond basically to form. So what you're going to do is you're going to bring another one of these molecules along, slot it in here, and bind this carbon effectively to this one here. So let me show this. So we'll bring in another molecule of isoprene here, okay, which we've sort of broken, you know, it's an intermediate, it's not isoprene anymore, okay, like so. So we'll bring another one of these intermediates in where we've broken these two bonds, forged this double bond in the middle here, but now both of these terminal carbons have free electrons. And this carbon here will also have a free electron, and basically we'll join them together. And of course this carbon over here will have a free electron as well. So we'll forge a bond here, okay? And then you can continue this process on and form massive great polymers. Okay, right. So basically, let me now show you the structure of the farnesyl group and also the geranyl group, and then I'll tell you about how you can add them onto proteins. So, we're going to draw the skeletal structures for these. Okay, so farnesyl is basically only going to have another, it's basically just going to be this thing that we've drawn here with one more added on. There's going to be 3 of them. Okay, so farnesyl, but we'll draw the skeletal formula anyway. Okay, so this is a farnesyl group that we're going to add on. Okay, so I haven't actually drawn you the skeletal formula for one of these polymers yet, so let's just try and understand this. Okay, so when we break the second of these 2 double bonds, okay, and forge a new double bond there, what is this molecule going to now look like? Well, it's going to look instead like this if we draw its skeletal formula. Okay, and of course both of these carbons now have free electrons that they want to join up. So let's go for that. Okay, so let's connect 3 of these together. So here we go, like so. Okay, so here's 1. Now what we'll do is connect this to another one. So we'll bring in another one here. So here comes another one of these isoprene intermediates, and we'll forge a bond between this carbon and this carbon, which we can do because they both have free electrons. We'll then bring in another one here, okay, like so, and we'll forge a bond between this carbon and this carbon again because they both have free electrons. Now, this final carbon and this final carbon here will still have free electrons. What we will do is we'll put a hydrogen on this carbon. So this final carbon over here will bring a hydrogen atom along with a free electron, okay, and we'll bind that hydrogen to that carbon and therefore saturate that carbon. Okay, so that's that end coped with. And now this carbon over here, what we can do is we're going to add this carbon onto the protein molecule basically. Specifically, we're going to add it onto cysteine amino acids. Okay, so I'm just going to break with the skeletal structure for a moment and put the methylene group here, which is this final carbon with these 2 hydrogens off. And then I'll put this single electron here to remind us that it still needs to be linked to something. Okay, and this whole structure now is called the farnesyl group that you've got added onto whatever this is added onto. So this is going to form a bond with something, and whatever this is added onto, you would then say that it has been farnesylated. Okay, right. So what we now want to talk about is geranyl groups. Okay, so geranyl groups, and then we'll talk about what you're actually going to add them onto within proteins. So, the geranyl group. Again, this is based on the structure of isoprene, and this time what you're going to do is you're going to polymerize 4 isoprene molecules together to get the geranyl group. Okay, so let's start this off. So we have one of these isoprene intermediates here where we have broken those double bonds, forged a new double bond, and now what we're going to do is take 3 more of these. So I'll draw them all out and then I'll show you how we're going to link them up. Okay, so here is the 3rd one along here, so here's that double bond, the methyl group coming off. Okay, and then one final one over here. Okay, so what we're going to then do is we're going to link this carbon to this carbon, because of course they have 3 electrons, this carbon to this carbon, they have 3 electrons, and this carbon to this carbon. Okay, so finally what we have is this carbon over here with a 3 electron and this carbon over here with a 3 electron. Again, we'll stick a hydrogen onto there to saturate it, so that's fine now, and then all that needs to happen now is that this final carbon over here, and again I'll break with the skeletal structure for a moment, it needs to link to something. Okay, and it's going to link to an amino acid residue on proteins, and then what you will say is that the protein has been geranylated. Okay, right. So, now let's see how we can actually add these farnesyl groups and these geranyl groups onto proteins. And when you add these onto proteins, you say that you've added a prenyl group onto proteins, or you would say that the protein has been prenylated. Okay, so let's see how we can add these to proteins then. So basically, you can add these on to proteins at their carboxylic acid terminus. Okay, so if we think about our polypeptide here, it's a polymer of amino acids, so it's an amino acid after an amino acid after an amino acid, etc. So you have lots and lots of amino acids. Now, you have 2 ends of the protein, 2 free ends. Now, one of them, the starting end of the polypeptide, will have a free amino group, okay? And the opposite end will then have a free carboxylic acid group. Okay, so this is what's known as the carboxy terminus of the protein, or the C terminus. Okay, and the end which has the amino group is then known as the N terminus, or the nitrogen terminus, or the amino terminus. Okay, so this is the N terminus. And it's often called the amino terminus because it's got an amino group right over there. Okay, so basically, you're going to add prenyl groups onto the carboxy terminus of proteins. Okay, now, basically, at the carboxy terminus of proteins, in order for you to add a prenyl group on, you have to have a certain structure there. Okay, and this certain structure that you have to have is what's known as the CAX box. Okay, like so, CAX box it's called. So basically, at the end of the protein, at the carboxy terminus of the protein, you have the final 4 amino acids. So let me show this. So what you'll have is 1, 2, 3, 4, and then right at the end you'll then have the free carboxylic acid group over here. Okay, so this final amino acid is denoted by this X here. These 2 middle amino acids here are denoted by the A's, and this 4th amino acid over here, so the one that is 4 along from the final one, so this is the final amino acid in the protein, this is the 2nd to last amino acid in the polypeptide, this is the 3rd to last amino acid in the polypeptide, and this is the 4th to last amino acid in the peptide. Okay, so in order to add prenyl groups onto the carboxy terminus of proteins, you have to have one of these CAX box structures. Okay, right. Now, why is it called a CAX box? Well, basically, this 4th amino acid here needs to be the amino acid cysteine, okay? And the single letter code for the amino acid cysteine is C, okay? So, it's actually this cysteine that you're going to add the prenyl group onto. Okay, so let me show you the structure of the cysteine amino acid as though it's in the protein. So I won't show you the structure of the pure cysteine amino acid, I'll show you the structure of a cysteine amino acid residue. Okay, so here's the amino group, and then here's the alpha carbon with a hydrogen coming off it. Here's the carboxylic acid group there. Okay, so it's as though it's bound in a protein. So this amino group would be bound to the carboxylic acid group of the next amino acid along, the fifth one up here. And this carboxylic acid group here would be bound to the this amino acid here, the third one in, okay, from the carboxy terminus. Right, so the R group then of cysteine is that you have a methylene group, and then off this methylene group you then have a phyl group, like so. Okay, and it's onto this phyl group that we are going to bind our farnesyl groups, or our geranyl groups. Okay, so let me, before I discuss the actual binding of the farnesyl groups and the geranyl groups onto this cysteine that's 4 in from the carboxylic acid terminus, let me discuss the CAX box in a bit more detail. So, what does A and A stand for? Well, basically, this tells you that in these positions you need to have an aliphatic amino acid. Okay, so a non-polar amino acid. So what key examples of non-polar amino acids are there? Well, let me show you some amino acids that would be considered aliphatic amino acids. Okay, so I won't draw out their full structures, instead I'll draw out their R groups. So basically, I won't draw out this core amino acid structure that's common to all amino acids. Instead, I'll just draw out this R group. So, for instance, in the amino acid glycine, you just have a hydrogen as the R group. Okay, and that's a very aliphatic amino acid. In the amino acid alanine, you have a methyl group as your R group. Okay, again, that's an aliphatic amino acid. On the amino acid valine, you have a structure that looks like this. You have a carbon, which then has a hydrogen coming off it, and then 2 methyl groups coming off it, like so. So this is the structure of valine. Okay, then let's look at the structure of leucine. Okay, leucine is basically just like a bigger valine. Okay, so it's got a carbon with 2 hydrogens coming off, a methylene group, and then one more along, it's got the same structure as valine, so a carbon with 2 methyl groups coming off, and then a hydrogen. Okay, so that's the structure of leucine. Another key example is isoleucine, okay, which is a isomer of leucine. Okay, and this has 2 carbons, like so, and then basically all you do is swap one of these methyl groups with one of these hydrogens off here. So you're going to put the methyl group off this first carbon, and then the hydrogen there, and then the final methyl group can remain where it is, like so. So this now is the structure of isoleucine. And these 5 amino acids, glycine, alanine, valine, leucine, isoleucine, they're some core examples of aliphatic amino acids that you could have in this these 2 positions here. Okay, so that's what you need to have in those 2 positions. And then finally, X, the amino acid that you have at position X, i.e. the final amino acid in your polypeptide, will determine whether your polypeptide is going to get farnesylated or whether it's going to get geranylated. Okay, so basically, if in position X you have the amino acid alanine, methionine, or serine, you're going to get farnesylated on the cysteine of the CAX box. And if in position X you have leucine, you're going to get geranylated. Okay, so let me discuss this. So, position X. If in position X you have the amino acid alanine, which we've already seen over here, alanine has an R group of methyl, okay, or if you have methionine, now I'll show you the R group of methionine. So methionine's R group is this structure here. It has 2 methylene groups, okay, followed by a sulfur atom here, and then off that sulfur atom you then have a final methyl group, like so. So that's the structure of methionine. Okay, and then the other amino acid that you can have in position X and get farnesylated is serine. So the structure of the R group of serine is that you have a methylene group then with an alcohol group off it. Okay, so if you have any of these amino acids in position X, what will happen is you will get farnesylated. Okay, so you're going to add a farnesyl group onto that cysteine that's 4 in from the end. Okay, so this will lead to the protein being farnesylated. Okay, right. Whereas, if in position X you have the amino acid leucine, okay, again, the structure of that we've already seen, it's over here, that will lead to the protein being geranyl-geranylated, i.e. having a geranyl-geranyl group stuck onto the cysteine at position 4 in from the carboxy terminus. And I should say that my terminology is not ideal here, because really you count the amino acids from the amino side, not the carboxy side. So really the amino acid over here would be amino acid 1, then amino acid 2, 3, 4, 5. So really I should say the amino acid 4 in from the carboxy terminus rather than amino acid 4. Okay, right. So let me now discuss how we're going to add these prenyl groups, or these isoprenoid groups if you like, onto the cysteine at position 4. So basically, if we've created a polypeptide that has this CAX box at the termination of its length, okay, so if it has the CAX box right at its end, then it can get a prenyl group stuck onto it. Okay, now how are these prenyl groups going to be stuck onto it? Well, we've got our structure of cysteine up here. So basically, if I just draw out that structure again, so here is the amino group that's involved in a peptide link, here's the alpha carbon, and here's the carboxylic acid group down here. Okay, we have our methylene group with its thiol group off, and basically we're going to take that hydrogen off the thiol group. Okay, so imagine breaking this bond, okay, and again this bond has 2 electrons in, one from the hydrogen, one from the sulfur. Imagine giving the electron from the sulfur back to the sulfur and the electron from the hydrogen back to the hydrogen. Now this sulfur has an unpaired electron. Basically, it's going to bind this unpaired electron with that unpaired electron that we saw in the farnesyl groups and the geranyl-geranyl groups. So what I'll do is I'll draw a farnesyl group linked off here. Okay, right. So this is going to look a little odd because it's suddenly going to go from molecular formula to skeletal structure. Okay, so here is the first carbon of the farnesyl group. So remember, farnesyl consists of 3 isoprene groups. Okay, so remember, isoprene groups now have this double bond here in the middle. Okay, so there's our first isoprene group. Then let's draw our second here. Okay, and then finally our third over here. And then of course we'll link this one to this one, this one to this one, and then of course put a hydrogen off that final one. So now what we have done is we've added a farnesyl group onto our cysteine amino acid 4 in from the carboxylic acid terminus, and the protein is now farnesylated. Okay, right. You can do the same thing for geranyl-geranyl groups, okay? And of course it depends which one you're going to put on is determined by which amino acid you have in position X. Okay, right. The one last thing to say is that this bond where you have the sulfur atom linked now to a carbon atom like that, that's what's known as a thio-ether bond. Okay, so this is a thio-ether bond here. Okay, right. Well, it also includes the fact that the sulfur is linked to 2 carbons. So basically it's like an ether. Okay, so an ether is basically when you have an oxygen sitting between 2 carbons, like so. Okay, so a thio-ether is very similar to an ether, except that you use sulfur rather than oxygen. And remember, sulfur is in the same group of the periodic table to oxygen, so it has very similar properties. Okay, so it forms two bonds, well, it can form more than two bonds, but here it's forming two bonds, and both of them are to carbons, so this is a thioether bond. Okay, right, so in the next video what we will turn our attention to is the fatty acylated proteins.